Hello to everyone joining us. We're just waiting for the attendees to load up. Sometimes this takes a couple of minutes. So once I see everyone's online, we'll start the presentation. Thanks very much. I think a few people are still joining us. I'm just going to leave that another minute and then we'll start the presentation. I think there's going to be a few more people joining us, but we'll wait for them to filter in. In the meantime, I'll just make the quick introduction. Welcome to ArrayJet's 2021 webinar series, where we're focusing on some of our customers' key applications to provide a bit of insight to the types of application where customers work, where they apply novel microarray technologies. So today, Shengsei Tao from Jiaotong University in Shanghai in China will be introducing his work, which will be protein microarrays for the systematic deciphering of the SARSCoV2-Specific humoral immune responses. If anyone would like to get in contact with Shengsei Tao, his email address is here, and he will be open to some questioning after the event as well. But before we go on to that, I'd like to give you a quick introduction to ArrayJet for those of you who are not aware what we do and who we are. We are a microarray solutions provider. We manufacture the quickest and fastest microarray platform on the market today. And we also offer services where we hire out our own scientists to be able to help you develop or run assays. So we offer a full range of solution from instrumentation all the way through to contract microarray services. We have a modular system that allows you to start research, relatively low volumes, all the way through to high volume contract manufacturing. And with our ArrayJet Advance, we work as both a CRO, so contract research manufacturer, doing basic R&D, all the way through to manufacturing large batches of custom microarrays. So ArrayJet, we work with our customers from initial contact through technical discussions, offering our own expertise, demonstration, and then eventually to hopefully to delivering instruments and after sales support. enabling our customers to develop cutting-edge technologies. And everything that we do, we try to work in partnership with our clients to add value to them. A quick introduction to Jiaotong University, Shengsei, and ArrayJet. So Professor Shengsei Tao joined Shanghai Center for Systems Biomedicine at Shanghai Jiaotong in 2009. Shengsei has worked with some very prominent microarray specialists, including Jing Cheng at Tsinghua University in China, and also with Heng Zhu at Johns Hopkins University, who's well known for his high impact work in high density protein microarrays. The Tao Lab uses a number of different microarray technologies to explore a range of different diseases, not only SARS-CoV, but also Mycobacterium tuberculosis. And if you want to discuss any of these things, please do reach out to Shengsei, and I'm sure he'll be willing to listen to any ideas for collaboration. So at this point, I'm going to hand over to Shengsei, and we'll start the presentation.
Okay, so I have to show.
Yeah, if you share your screen now, and you'll be able to take over from me.
Okay. So can you see my screen?
Yes, we can. Yes, perfect.
Okay, great. So let's get started. So first, I want to thank Ronald for the nice introduction. As he introduced, this is Seng Tao from Shanghai Jiaotong University. So today I will going to show what we have done in the past 1.5 years on COVID-19 by using the microarrays, both protein microarray and peptide microarray. So this is basic information about COVID-19, which I got by yesterday. So till today, there's more than 179 million people have been infected around the world, which have already caused about 4 million deaths. And the good thing is, like, we have a lot of people, vaccination now is accelerating. A lot of people have been vaccinated, hopefully, which can like change the current situation around the world for COVID-19. So if we talk about COVID-19, actually it's caused by a virus called SARS-CoV2. And this virus is RNA virus, which have a genome about 30K. So in total, they encoded about like 28 proteins, which include 4 structured proteins, 8 accessory protein, and 16 non-structured proteins, which is called NSP. So among these proteins, the most important protein is the so-called spike protein. The spike protein actually is the important because it interacts with a receptor on cells called ACE2, which mediate the attachment and infection of the virus to the host cells, thus which is key for infection because the importance. So as protein now is a key target for therapeutic antibody development and also as well as vaccine development. For most of COVID-19 people, they are recovered by their own immune system since there's no, even till today, there's no approved effective drug for treating COVID-19. If we talk about immune system, they include both B-cell immune response and also T-cell immune response. For the B-cell, actually sometimes we call it humoral response, which include a lot of different antibodies. For example, like IgG, IgM at different stage. And also there are various different cases. The reports have shown covalent plasma could be used for potentially used for treating COVID-19, and the major effect factor, what we think is antibodies. So actually COVID-19 is a disease. If we're talking about disease, there are 3 key steps. One is the prevention, the second is diagnostic, and third is therapeutics. Among all these key steps, as antibodies all plays very important roles, like for new vaccine, new biomarkers, and also new drugs. So our goal is to try to decipher the global picture SARS-CoV2-Specific antibody response. So if we talk about antibody response, actually on a global view, which is there's some question we have to ask. The first question is like, since there are so many proteins for this virus, which protein which is most immunodominant. And if we talk about S protein, then the people will be interested to ask which part of S protein, like which of the epitope is more important for the B cell immune response. So this is said by a Chinese Kongzi 2,500 years ago. It's called 公与圣其实必先利其 in Chinese, but it's translated into English called, if you want to do a good job, sharpen your tools first. So for this study, we have two tools. One is protein microarray. The second one is a peptide microarray. For protein microarray, as just Ronald have introduced, our lab have been working on protein microarray technology for many, many years. So we fabricate a lot of different protein microarrays and do a lot of different applications, as well as doing technology development. So to answer the first question, like which protein is more immunodominant, then what we have to do is we have to clone and express the proteins. So back to last year, January sometime, we start to construct all the expression constructs. And finally, we have been successfully purified 18 of the proteins. That's for the first version. And the second version, we got like 21 out of the 28 proteins. And we can see most protein, we have pretty nice signal. So after we have the protein, then we have to print the array. And here it comes with the array jet printer. So basically we use this printer to print about like over 1,000 slides for the protein microarray as well as the peptide microarray. I will talk about in a minute. So after we have the proteins, then use array to print the array as what we showed in the right side. And on here, we can show each protein we have like 4 spots for the replicate. We can show the array is pretty nice, as well as the quality of the array reproducibility is also good. After we have the array, then we use one covalent sample, which is from COVID-19 patient. We probe the sample on the array. We can see pretty nice signal for some of the proteins. And while for the control, there's no very weak signal could be observed. So after we have the array, then the first thing is we have to collect the sample. And luckily we got some sample from Guangdong through one of our collaborators. This is a small cohort which include 29 sera from covalent patients. As well, we also have some control group samples, like different diseases like cancer, and also we have some health control. So we probe all these samples one by one on the protein microarray that I just showed. So after we finish the experiment, then we collect all the data from the microarray doing the data analysis. And based on the data, we generate a heat map for both IgG and IgM. For here, what I showed is IgG profile. So we can see from here, it's like this is heat map. We can see basically the microarray could be used to differentiate clearly for the COVID-19 patients from the controls, as what we show here. And the most significant signal is observed for both M protein and S protein, which is also consistent with many of the other studies. While for the control part, there's no signal, very weak signal could be observed. So for the IgM profile, it's similar. We also can use the microdata to easily to differentiate the patient from the controls. And then N protein, S protein also the strongest. The difference, like for the IgM signal, is a little bit slightly weaker than the IgM, which I think is understandable because the sample that we test on the array is a covalent sample. And as the people already know, for this at this stage, IgM level could be much lower than the initial stage. So this is, we go detailed to see which protein is more important. And then we focus on two of the most significant protein. One is S protein. The second is the M protein. So for here, I show the left panel on this image. So just for one example, this is S protein, the S1 subunit with spike protein. the left part is from the patient, the right part is from control. We can see based on the microdata can clearly differentiate the patients from the controls for all the proteins. They're similar, and we have different version of S protein, both S1, S2, and RBD. The red part I have to point out is a protein that we collected and prepared from eukaryotic. So much better signal is observed for this part. Our explanation is that maybe this signal, better signal is due to the glycosylation, which is it's possible when we collect prepared protein by using eukaryotic system. So M protein got similar signals. So the point for here is like both M protein and S protein could clearly separate the patients from controls, and maybe the eukaryotic prepared protein is better. For IgM, we got similar results like to that of IgG. So initially, the goal for this study is like we want to see the whole global picture for the B cell immune response, not just S protein and N protein, which is expected. After the profile, also we found some protein which is unexpected, like ORF9b, and NSP5 also have a higher immune response among the patients, even though not all patients, but for some patients, the signal is pretty high. And by performing the differentiation analysis, we can see also both these two proteins can differentiate the patients from the controls, as what we show on panel B and panel C. So in another part, we want to try to see whether there's any correlation between these two proteins and the M protein and S protein. And what we found here, there's the correlation is not good. So that means the immune response to ORF9b and NSP5 maybe is independent to that of N protein and S protein. So that's the point for this part. And after we have the microarray data, then we want to see, and back to that time, last year, a lot of people is highly interested whether this signal is also related to clinical parameters like gender, age, and some other important parameters. And then we perform the analysis. What we found here interesting is like the IgG signal, especially for S1 protein, which is positively correlated to age for both male and female. And for female, much better correlations we observed. That's for the age part. And also for one of the key clinical parameters called LDH, lactate dehydrogenases, which is also positive correlated with the antibody signal, IgG signal to S1. And for another parameter the lymphocyte ratio, which is negatively correlated with that of the IgG signal, which is also very interesting. After we got the paper published on Nature Communications, various different reports have also confirmed this discovery. So this is for the protein microarray part. So after we finish the first small cohort, we make sure that our system works and works pretty well. So then we start collecting more sample. Luckily, we found a collaborator from Wuhan, Huazhong Science and Technology University, Professor Xiongling Fan, and he has a lot of sample. Then we collaborated on a large cohort, which include more than 3,000 COVID-19 samples. Also, we collect negative controls, about 600. So we follow the same procedure. We perform the large-scale analysis for all these samples on the protein microarray that I showed. So for here, because we have many more samples, then we got a much clearer picture about the B cell, the antibody responses for both IgG and IgM. For here, what we show here is for panel A, the left part is for M protein. This is the S1 subunit of S protein, M protein, and N terminal. That's N terminal of M protein. We also got C terminal M protein, which found all these proteins have very high response rates. For the number shown here is the so-called response rate. That means if you, for this number like 96.7, that means if you have 100 people, 96 of them, more than 96% are positive. That's pretty high prevalence for this immune response for S1 proteins. And beside S protein and N protein, many other proteins also have clearly response to some or some to many of the patients. Like some, for example, like one for here, NSP7, which have pretty high prevalence of positive immune response for against these proteins, and RDRP also there's a pretty high. Another one for here is like ORF3B, pretty high. So based on this study, we can see global clear, more clear like a picture about the IgG response against COVID-19, SARS-CoV-2. That's for the IgG part. For IgM part, it says signal is similar. It's just the signal is much lower than that of the IgM. So for here, we got a much clear signal and got a bunch of paper published on this part. So this is for the protein part. Then if we go deeper, we want to ask second question, which part of the S protein, which is more significant immunodominant for the IgG response. So then we come with a second tool. This is a peptide microarray. So you or maybe all know that for the spike protein of SARS-CoV-2, there is the total length is about 1,273 amino acids. So what we did is we start from N-terminal of this protein. We designed the peptide like 12 amino acids. So for each of the two adjacent peptides, they have 6 amino acids overlapped. So in total, we got 211 12 more peptides. Then we performed the synthesis by our company, and at last we got 197 then successfully synthesized and also soluble. Then put all these 197 peptides as well as some of the controls on the microarray. This is for the first version of the peptide microarray. We also test 3 different concentrations. So this is to show you how good the quality of the peptide microarray. After we have the microarray, then we want to test whether this microarray could be used for the COVID-19 related studies. The first application is like we click some of the antibodies and test antibody on the microarray. And for here, just for one of the example, we can see the antibody signal, which is clearly and very specific to some of the peptides. That means this antibody may be just specific to one of the epitope. That's for this one. The second one, we have a collaborator in our institute. He is developing some kind of mRNA vaccine. After he inject the vaccine to the mice, they collect the serum. Then we analyze the serum also on the micro. We can see the vaccine indeed induce some of the specific antibodies, which is against to some epitope of the S protein. This is for the second application. The third one is this one we want to do is to test the COVID-19 serum on the protein microarray. Then we got a bunch of signal for both IgG response and also as well as IgM response, as what we show here at this corner. So by doing this, we can make sure the quality of the array is good, and also it could be used for very different COVID-19 related applications by using this peptide microarray. So then we use the same set of the large cohort that we collaborated with Professor Fan Xiongling from Huazhong University of Wuhan. So we probed all these samples one by one on the peptide microarray. So after we got all the signal, we want to see and use the global view. Then what we did here is we put all the signal as spotted all the signal like this. So this is exactly we aligned to the protein sequence. Like this is S protein sequence, different domain. So for each spot represent one signal against this peptide from one patient. So overall, then we got an overall picture for the patient part. And also we have about 500 of the control samples. If you compare this part to this part, we can see there's the signal we obtained from the patient part group is very specific. Then if we look deeper, what we found, some unexpected observation we made. The first one is like for the RBD domain, which is so have very highly immunogenic for the immune response. Actually, we unexpected, we observe very few signals for the linear epitope, except a few of them. And also we observed some like hotspot for the B-cell immune response. One is called the CTD, C-terminal domain of the spike protein for the S1 subunit. And another hotspot is, for example, is the area which is close to the so-called fusion peptide region. So this is also unexpected. And for the RBD part, our explanation is that maybe the RBD is the epitope is highly conformational epitope enriched, not linear epitope. That's possibly one of the reasons. So yeah, and also we performed analysis, try to see whether the signal immune response is also correlated somehow to the glycosylation part. And just based on look at this image, we cannot see there's a high correlation between glycosylation and the immune response. So after we got the overall picture, then we focus on some of the peptide we define as significant peptides based on the response frequency. So we clicked 16 of them, and the last 3 we clicked is not that high, just because what we tested, because there are from the RBD domain, which is also functionally important. Then we, in total, we select 19 of the so-called significant peptides. If you look at, check our data with that of the other research studies, we found for some of them they are consistent, but also we identified many of the new highly immunogenic peptides from the S protein, spike protein. So then we want to see whether these significant peptides are located on which part of the spike protein. By looking at their structure, what we found that, which is not unexpected, many of the over the peptides, they either located on the surface of the trimer of the spike protein or located on the monomer of the spike protein. That's unexpected, okay? So then we want to validate the specificity of the antibodies. What we did is we used some of the peptide to enrich the protein from the SIVA, then also use the array to check their specificity, also works pretty well. And the panel C just show you one of the epitope, where they're located on the surface of the spike protein. So for that like analysis, what we performed homologous analysis, because we know there are some other like coronavirus, which can also infect human, like the famous one like SARSCoV, MERS, and some other like OC43. So we perform a homology analysis for this significant peptide. For here, just for one of the examples, like for the peptide S1-76, 77, 78. What we can find here is like this area is highly homologous between SARS-CoV-2 and SARS-CoV, and also as well as to one of bat coronavirus, but not highly homologous to other like coronavirus. So at the last step for the protein marker part, we want to see whether this peptide can trigger antibodies which possess like neutralization activity. Then what we did is we use a peptide to enrich or purify peptide-specific antibodies from the patient sera. Then we perform, after we got the antibodies, we test their specificity on the peptide, and also then we performed neutralization assay by using this enriched antibodies and on a pseudovirus model indeed, we found 3 of them, like S193, S1105, and S278. They do have some neutralization activity. That's for 3 of them. So this is for the protein migrate part. And in one of the last experiments, like because we want to test all so-called significant peptides, but we don't have enough patient serum. So what we did in this part, like we synthesize all these peptides again and conjugate them on KOH and then inject the mice. Then we collect serum from the mice. We tested the specific of the antibodies that are triggered by using the peptide microarray. You can see for most of the peptide, we got significant antibody response. And after we collect all this antibody from the mice, then we perform the authentic radius assay. And here, this is control, is neutralizing antibody. What we found here is like for some of the peptides, they can induce some of the neutralization activity, but it's just weak. It's very weak. So this is basically what we found by using the protein microarray profile, the peptide microarray profile, this kind of like a study. So then we got the results. So what we conclusion for here is like all the peptides could trigger specific antibodies. So, and also we found that for the peptide, their antibody level is proportional to the number of immunization, like especially of the 3rd and the 4th immunization. So also we observe some weak neutralization for some of the peptides. So this is for the protein, the peptide part. And after this, for diagnostic purpose, then we also perform analysis, try to see is there any protein peptide could be used for biomarker for diagnostic. And indeed, we found some of the protein like N protein, S protein, which is not a surprise. Surprisingly, we found some of the peptides, even for single peptide like S278 or 12 more peptide, have pretty nice capability for differentiate the COVID-19 patients from that of the controls, like what we show here, S278 peptides, which is pretty well, can pretty well differentiate the patient from the control. And for this peptide, we are now is under developing, we collaborate with another professor in John University, try to develop a diagnostic kit by using this peptide. So overall, during this time, I want to convince you guys that we have generated a global picture for the antibody response of COVID-19 specific antibody response. So the first question we want to ask, which protein, which is immunodominant, and then is which area of this protein immunodominant. So we, by using 2 tools, one is protein microarray, the second is a peptide microarray. So if we put all this, our data together, then we have this image. If you guys miss all the other part, I urge you guys to remember this part, this slide. So what we did is the sample include all kinds of COVID-19 samples from asymptomatic, mild, severe, critical, days recovered, and even vaccinated that we are doing now, which is include like around like 40,000 samples. Then by try to generate, construct a global picture of the B-cell immune response. We use different tools, protein microarray, peptide microarray, from protein level and peptide level to generate the picture. And also we have another technique, but I don't have time to address this technique today, which is the high super epitope map technology. We can also map the epitope to amino acid resolution. After all this, we can now confidently say we have a global picture for the B-cell response of COVID-19 patients. So based on this response or the global picture or the landscape, we can then address going to vaccine. This is something we are doing for diagnosis and also for therapeutics, which is to guide people to design neutralization antibodies. That's for this. So you can see for all this work, we have to collaborate with other people. If without help from other group or other people, which is totally impossible, especially during the COVID-19 crisis last year when in China. So there's a lot of people I have to acknowledge. The first is the people in my lab. There's most of them are doctors. And also my collaborator, Professor Yin Tianyan, and his student, Xiaolong Tian from Fudan University, and my collaborator, Professor Xiongning Fan, and his student, Qing Lei from Huazhong University of Science and Technology, and other like collaborators, like Mr. Jie Zhu from Foshan Force Hospital, and other collaborators as well. So here's it's like the almost a final slides. It's a core team back to last year. It's about March sometime. By when we decide to perform the analysis for 3,000 samples, then we ordered over 1,000 slides from United States. This is the when we got the slides. Then the people are very happy, so we take a picture for this one. And here is some of the publications we have already published by using the both the protein microarray and the peptide microarray for doing the B-cell immune response analysis. And here is my contact information. If you have any question you want to ask in the future, you can just send me an email simply, and I will try my best to answer your questions. Thank you all for your attention.
Thanks very much, Shengse. We have a number of different questions. I'm just going to share my screen quickly.
Yeah.
Yeah, we've got a number of different questions from several different sources. So I've got a few questions, first of all. So are these arrays affected by the new spike variant proteins?
What?
Are these arrays affected by the new spike protein variants? Or would you need to adjust that?
I think so. So but we still haven't tested that yet. So we have a plan to include some of the most famous variant on the array and update the microarray to maybe the third version. So, but we haven't run any test on this microarray by using, the people which is infected by the most famous variant, like the urea variant, the P1, and someone from Brazil, this sera, but we haven't run this yet. The problem is like in China, we don't have that kind of sera to run this kind of test, but we will do that if there's, necessary.
Okay. Yeah. And have you done any studies into the difference from responses from convalescent patients and compared to vaccinated individuals?
Yes, we do. So we have another paper which is under review, which is by doing that we compared 3 different groups. One is convalescent patients, another is patients. And the third group is the vaccinated people. So we do observe some difference. And actually overall, their pattern looks very similar, but we observe some difference for some of the protein, which is quite interesting.
Okay, so they're relatively...
Probably in 2 or 3 months, yeah.
Okay, cool. I'll be really interested to see that. So I'm now just going to move to the Q&A. I'm going to start at the top. So what part of the process, what was the most difficult part of the process, and what have you learned from this that you would do differently, even when the next pandemic arrives?
So this is a question from Ian, right?
Yeah, this is Ian, yeah.
Yeah.
So, a typically difficult one.
It's very, I think it's very good. So the most difficult part, I think there are 2 parts for the micro study. One is how well I get proteins. So because, for SARS-CoV-2, because the genome is not that big, but if there's other like a pathogen, they have thousands of genes, which is very hard for you to prepare the proteins in a very short time. For SARS-CoV-2, just because I have 28 or 27 proteins, then we have we are able to, prepare the proteins from gene synthesis to get the purified protein just in 1 month. We can do that. That's the first part, I think, which is very important. The second part for this kind of assays, like the sample, where you can get high quality and a large amount of sample. That's the key. I'm kind of lucky for the first small cohort because I have a long-term friend at Guangdong Foshan. He's a director of the hospital, so he have samples. The second one is sorry. The second one, the large cohort is just, I share the reagents on the website, and then we found a collaborator. Before we start the collaboration, I even haven't met him for once, but we just met it by this, kind of collaboration. So this is I think the key for the second part. So the question is like, and if for next pandemic, I think the things like the scientists always have to get ready. So for us, I think even we have another like a pandemic, which is similar to COVID-19 or, or similar, we can do that in much shorter time than for here, because we have learned a lot during 1.5 years, the past 1.5 years. So we have some experience. Also, we, made some like something which is not right when we do it at the first time, but next time we will do it much better. I think so. Yeah. Good.
Thanks very much. And then the next question is from Ryan Liu. What are the processing? What is the sample processing you did before applying to the microarray? How did you get the samples in the correct format?
That's and then also kind of a technical question.
Yeah. Do you want to answer that directly, Ryan?
I can see his question. So 2 questions. So the first question is like, what's the sample processing procedure, right? So for here, it's pretty simple. When we profile the serum on the microarray, the typical is what we did is like, well, after we have the serum, then we do some kind of deactivation, because even for the covalent serum, we worry about the infection. So this usually like a 56 degree for half an hour or 1 hour. That after that, then what we did is we just like dilute the serum. Usually it's like a 100 or 200 times dilution in PBS buffer. Then we incubate the diluted serum on the microarray. And after that, you can do wash, and then you come with the second antibody, which is an human antibody, but it's some kind of like a fluorescent conjugate, either CL3 or CL5. Then after that, that's a typical microarray procedure. Then you can, after you wash the slides, you dry it, then you can obtain your signal by using a microarray scanner. That's for the first question. The second question is like, we have long-term experience by using a very different kind of microarrays. We also have an older microarray, which is from a domestic company in China. It works pretty well, but just because it's just too old. Then for most of the COVID-19 microarray that we fabricated, what we used is the originated microarray. We just obtained like 2 years ago. So it works pretty well. The variation, there are still some variations, but I think the variation is almost neglectable. So it should be okay. We check reproducibility for all printings. It works really, really pretty well. We think that's not a concern for the spot variation by using the originated microarray.
Thank you very much.
Ronald.
And then the next question is from Adriana at Inopsis. So you mentioned the diagnostic kit. Would that be in a microarray format, or is that going to be in another format?
Okay, that's a good question. So when we perform microarray analysis, we usually use microarray as some kind of screening tool to identify some biomarkers, either protein or peptide, or the combination of protein and peptides. But after we have the result, usually I don't think we will use microarray format to to develop the diagnostic kit, which is not well suited for traditional applications. So what we did is now we're trying to use the peptide like S278. What we did, we want to develop some kind of like a handheld or portable device that people can use this at home or just by themselves or point of care. That's something we are trying to do. And also the overall form will be suitable to most of the hospitals in China and also in other countries. But for Microarrays, we cannot do that.
Okay. And a follow-up question to that, what was your opinion of using microarrays within diagnostics?
That's really a good question. For microarrays, for diagnostic, I think that people have been trying that for a long time. So, but to my experience, I think it's still there are very few success. The thing is like if you use traditional microarray, like the fluorescence based, I think it can only be, the test can only be run in some like, laboratories, which is well equipped laboratory can only perform the analysis. But if you want to put the kit, microarray-based kit to hospital or even to personal use, which is traditional micro format cannot be doing that. But say if you only have a few of the proteins targets, you can make a small microarray. Then if you can develop some kind of like a visible kit by some kind of color, then maybe you have some chance. Like I know there's a company in China called Digital Health something, which is have a very like only like C12. I remember is for cancer. They can for the diagnosis of 12 cancer biomarkers. Then what we did is they just put big spots, much bigger than the micro, but they also call the micro, 24-well format.
Yep.
So then also give some kind of like a visible assay. It's similar like ELISA. Then we put this use for the diagnostic of some, like it's not diagnosed, like some kind of screening for the cancer biomarkers. It works pretty well. And then I think they made a lot of money based on their product. That's one example I remember works pretty well for some kind of clinic applications.
Yep, well, this is a challenge that we see on a regular basis. If anyone would like to discuss that with us, we're more than willing to have a chat. But yeah, thanks for your summary. Thanks for that. Yeah. Another one from Mark. What technique are you planning to use for the amino acids level epitope mapping?
Yeah, so the thing, because today we have only like 30 minutes, right? And because already is doing microarray, so I just majorly talking about the microarray. For that part, we have a technology called AB map. We have been spending like in the last 5 years. That technology, we already have some paper published. If you want to know more, I can send this paper for this.
Yeah.
This Mark, right? I can send you the paper if you want to see that. Yeah, we already published. For that one, I can in brief. It's a combination and also generation sequencing. We have developed a technology which can be used. I don't know how to control switch.
It's okay, not a problem. Don't worry.
Sorry for that.
It's okay.
It's not intended. So yeah.
Yeah. Okay. I can make an introduction to.
Okay. So that's called AB map. So we have a combination of phage display and next generation sequencing. We have the technology which can do the amino acid epitope mapping, which is so far we have update to version 3. We are managed to do that by 3 people in 1 month for 20,000 antibodies. So it's pretty powerful. We will have some other paper which is in preparation on that technology.
Fantastic. Mark, I'll send an introduction to Mark so that you can contact him directly and find out a bit more detail.
Mark, send me an email if you want to see more. Right.
Brilliant. So I've just got a couple more questions. Which applications are you most excited about for this array?
for the SARS-CoV-2 array?
Yes, yeah.
You know, initially what we want to see, we want to try to identify some like epitope, which is outside RBD, which can trigger like a high potent neutralization activity, but unluckily, unfortunately, we didn't observe any of them. And this is kind of like a disappointing, discard, but also it's confirmed. It should be useful for the audience, the community for vaccine design or neutralizing antibodies. And so far, I think the most interesting or promising part for based on this, the Microarray results are two. One is for the diagnostics, which is a kit we are developing now. Another one we are trying to, based on the epitope that we have mapped, try to develop a new way, a new technology for vaccine development. This is also is not an ongoing project in my group. That's one of the most interesting part. I think it's very promising. We are doing that.
Brilliant. Finally, what if I would like to use this array? Is it available for others to use?
Yes. So in China, it's available. We have already collaborated with a lot of people around China to doing the analysis, and also we have already licensed this array to. There's a company called CDI, in CDI laboratory in the United States. I think they have already provide a lot of service in the United States, maybe also in Europe. And another way, I think I have already been talking with Origin and Ronald for a few times. And if the people from Europe want to use this array, maybe we can start some kind of collaboration through Origin. Then we can do that.
Yeah, I would echo that. If anyone is interested,
Yes.
please let us know, and we can do some investigations.
Yes. Yes.
I'd just like to say thank you very much, Xing for an excellent talk.
Yes, it's my pleasure.
Yeah, and just congratulations on getting something in place so quickly. I know when we discussed at the beginning of the epidemic last year, your response was very quick, and it was such a short period of time that you managed to get something in place and start creating valuable data sets that have been used for some really interesting publications. So congratulations, and thank you very much for giving us an insight into your work.
Okay, yeah, no problem. Yes, really my pleasure. I think this is the responsibility as a scientist. We have to do something, right?
Yeah, I agree. And to echo what you said before, I think the way you were able to do this is through collaboration and connection. So that it's vitally important to introduce people and to provide the network for people to understand what's going on in other parts of the world.
Yes, that's true.
Okay, so finally, I'd just like to say we are going to do another webinar. This is going to be at the end of July. The date's to be confirmed, but what we're planning to do with this webinar is to work on, to give an introduction, Microarray 101. And it's going to be hosted both by ourselves and also our partners at Grace Biolabs, who manufacture nitrocellulose slides and surfaces, amongst other surfaces as well. And also our partners at Anopsys, who manufacture scanners that allow people to scan the slides and generate the data. So we will be announcing this in the next couple of weeks, and it will be available on our website to register with. But I'd like to thank everyone again for attending, for the brilliant questions, and we look forward to seeing you on the next webinar at the end of July. Thanks very much, everyone. We look forward to seeing you next time.